Hello and welcome to the webinar. This presentation is titled Advanced Technology for Broodstock Management and Research. This webinar is sponsored by Biomark and hosted by Hatchery International. My name is Marilyn de Guzman, editor of Hatchery International, and I will be the moderator for today's discussion. I'm excited to introduce our panel of experts today. But before we begin, just a few housekeeping reminders. You, the audience, will be in listen-only mode. However, you will have the opportunity to post your questions and comments to any or all of our speakers by typing them under the Questions tab on the webinar panel. The Q&A will commence following the brief presentations by our panel speakers, but you may post your questions at any time during this event. This webinar is being recorded and all participants will receive a link to the recording following this event. It's now my pleasure to introduce our speakers today. First up is Bert von Siebenthal. He's the R&D manager for Valperca, based in Switzerland. We also have Dr. Kenneth Overturf. Ken is a research geneticist with the USDA Agricultural Research Service, and he's based in Hagerman, Idaho. And last but not least, we have Hayden Bliss. He's the Director of Sales and Marketing at Biomark in Boise, Idaho. Welcome, everyone.
Morning.
So now at this time, I will turn it over to Hayden to start off the speaker presentations. Over to you, Hayden.
Excellent. Thank you, Mary Lynn. And I just wanted to say a quick thank you to everyone on the webinar joining us today. I hope that this finds you and your loved ones and your families healthy and safe at this time. So, and I also want to thank Bert von Siebenthal with Valperca in Switzerland and Ken Overturf with the U.S. Department of Agriculture for joining us today as our expert panelists. We appreciate your time. We hope you find this webinar informative, and please ask questions throughout. We've got a lot of great information for everyone today. So hope you enjoy it, and thanks again for taking the time to join us. And a little bit of background on me. Just really quick, my aquaculture journey began at the university level. I had a number of courses in aquaculture and fish rearing at the University of Idaho. And I also worked on our on-campus lab at the university, doing some work for some of my professors in the laboratory. After that, I went to the Idaho Department of Fish and Game, where I worked with salmonids, and then came to Biomark as a biologist, working on our fish tagging and field programs, and currently work as the Director of Sales and Marketing. I spent several years living in Europe, working with aquaculture customers in Europe. So a lot of work done in the UK, Ireland, Norway, Central Europe, and then in the Mediterranean as well. So, and then back here in the States, North America, the US and Canada, have worked with a number of aquaculture companies. So my background in aquaculture comes primarily from the industry side and supplying technology to aquaculture companies. So just a little quick history on Biomark. We've been around for over 30 years. As some of you may know, we supply RFID technology and solutions. We commonly refer to those as pit tags in our world. And we've worked with programs, as I mentioned, I've worked with programs all over the world, everything from salmon, tilapia, catfish, shrimp, sea bass, sea bream, and the list goes on. And one of the primary focuses, as I mentioned, of the technology that I work with and that we provide to breeding programs is on the identification side. So one of the cornerstones or the foundations of a breeding program is quite literally being able to identify each individual in the program. So the pit tag technology is a great way to do that. And to take it a step further, once each individual has been identified, then we're looking at taking DNA samples. So providing solutions for the semi-automation of genetic sample taking. And then, one more step in that process is then, okay, now we handle the fish. We need to collect data from the fish, whether we're tagging them, whether we're taking a genetic sample. So the automation of that data, so reduction of transcription errors, and then, compiling peripheral devices, a measuring board, a pit tag reader, a scale, those sort of things that can collect all that data, put it in one place, and provide us with robust data that, breeding managers can make management decisions on, essentially. So that's a quick background on myself and Biomark, and I'll turn it over now to Bert von Siebenthal with Valperca in Switzerland.
Thank you very much, Hayden. Hello, everybody. I also want to quickly introduce myself. My name is Bert von Siebenthal. I'm Swiss. I'm a biologist, and I have a major in zoology, but I specialized in fish during my PhD. After the PhD, I decided to stay in the fish world, and I then worked for 8 years at the Center for Fish and Wildlife Health. This is part of the University of Bern in Switzerland. At the center, I was in charge of the fish disease diagnostics, but I also was planning and supervising research projects within the context of fish health and fish welfare. This center also included the Swiss reference lab for notifiable aquatic diseases, and so we also did the diagnostics for these diseases there. Additionally, I worked for 8 years at the Federal Food Safety and Veterinary Office, and there I was also in charge of all kinds of fish welfare, fish health, and aquaculture inquiries. And since November 2018, I work now for Valperca, a big producer of European perch. And all this together gives about something more than 10 years of experience in the industry. And what I think is really interesting for me is that I got to know the industry from 3 different perspectives. So the veterinary perspective, the authorities perspective, and now the producer's perspective. And this quite helps sometimes. I would quickly like to talk a little bit about the company I work for now. Like I said, it's called Valperca. And we are basically the largest producer of European perch, Perca fluviatilis, in Switzerland. And we are also among the world pioneers for perch production and recirculation aquaculture system. The company consists of 2 facilities. It's the hatchery called Percitech, which is in Chavornay. This is in the French-speaking part of Switzerland. And then we have the grow-out facility, which is in other canton, about 2 hours away. In the hatchery, we have 8 different broodstock batches with slightly shifted temperature and photoperiod management. So this allows us for a year-round production of fingerlings. In the grow-out facility, we produce momentarily about 485 tons of fish per year. This means about 170 tons of fillets plus other products made out of fish meat. In Radon, we also have our own little processing plant where we produce the fillets, fresh and smoked, but we also produce various dairy products like tartar, several sausages, nuggets, and other products. You see on the right side, La Perche Loe, that's the brand name that we use to market our products. You can Google us and then you drop on our website. Unfortunately, it's in French and German only. Now a little bit about my role in the company. I'm a member of the board, but I'm also the R&D manager. And there in this function, my general task is the constant improvement of the rearing conditions and procedures. And yeah, when I started this job about 2.5 years ago, my boss gave me a list with about 150 subjects to improve. Now, after 2.5 years later, the list contains about 200 subjects. So you can see this is really a constantly evolving thing that will never really end. But nevertheless, we have about a couple of main fields that we constantly try to improve. And one of them is the improvement of the perch feed. There is up to now no real perch feed available. So we are trying to develop this together with some feed producers. And we are also still trying to improve the feeding strategies. Then the important thing is also to make the production more sustainable. And of course, the establishment of a Broodstock Selection program, which is the topic of that meeting. Just, yeah, you can see that My role is more the role of a generalist and coordinator than a specialist for a individual field. Just if you wonder how these fish look like, if you're not familiar with European perch, that there on the right side you can see it. And something really particular is that these fish don't lay individual eggs, they lay egg bands that can be 2 meters long. There on the top picture, you see one of these egg bands. That's a picture I took in a Swiss lake. And this characteristic of the eggs makes the artificial fertilization a bit more tricky than with other species. That's it about my person, and so I would pass over the mic to Ken. Thank you.
Good morning. Hi, I'm Ken Overture for the USDA ARS. Showing where I'm situated here in southern Idaho. Next slide. My background a little bit. So I did my PhD work in cell molecular biology, some postdoctoral training in medical and molecular genetics, and a brief stint down at the University of Alabama. So I came by my way of working with fish later on in my career, but currently since then, I've been 21 years with the ARS. As a research geneticist, I'm stationed at the Hagerman Fish Culture Experiment Station here in southern Idaho with my collaborative research partners, the University of Idaho, the Aquaculture Research Institute, and the Columbia River Intertribal Fish Commission, which makes us a really good group, both federal and academia, able to look at aquaculture long and short-term projects and the conservation side of fisheries. So, go on. Now, the reason we're stationed there in southern Idaho, the ARS is about 70% of the commercially reared rainbow trout in the U.S. is reared about within 40 miles of our facility. So the main focus of our group, which contains several ARS scientists, is reducing primary costs and optimizing production. All right, and we do this through developing genetically improved strains that we can release to the industry, ingredient testing and improving dietary formulations, and we do a lot of environmental monitoring. So we know specifically the water conditions, the effect of diets on conditions, the effect of diets on fish, and these interactions. Then the whole idea is to put this together into progressive management practices, because when everything all fits together is when you can have the greatest chance of improvement. On a basic side, for the research that we do, specifically, we look to identify the biological processes responsible when you select for specific phenotypes and what other changes occur in the animal, all right? And some of these we look at specifically are dietary utilization. I'll address that a little bit further. Nutrient processing, pathogen resistance, and again, water quality adaptation. So, move on. Now, so we currently maintain 2 lines of selected rainbow trout, and these are in the selection for utilization of sustainable diets. Now, for those of you not aware, in the past couple decades, most fish have been reared on a diet heavily containing fish meal and fish oil, and these are finite products, and they were utilizing most of them. With the rapid growth of aquaculture, these products they're non-sustainable, and the prices went up, and here in the past decade, there's been a very extensive amount of research, both by industry and scientists, to modify or change the protein and lipid products utilizing these diets so that they are more sustainable. The issue with that has been that sustainable protein products typically come from plants. The utilization of plants, when you include them at high levels, has a lot of negative impact on fish health and growth. So we've looked for selecting fish to utilize these and determine physiologically the changes that occur in the fish when they are selected. And just shown here on the side is one of our selection programs started back in 2002. And as we've increased the stringency of the diet, and that's by including higher levels of soybean meal in the feed and selecting for performance, we've been able to generate fish that grow better and do not develop intestinal inflammation or enteritis, as it's termed, in the selected fish as opposed to non-selected fish. And then we also evaluate these corollary changes that I mentioned before that we find linked to immunomodulation, amino acid transportation, muscle accretion, and pathogen resistance. And finally, I guess my role here is some of the work that I've done, obviously in our program with our Broodstock and our genetic selection programs. I've also worked with a number of companies, both within the US and internationally, on either auditing or evaluating their current broodstock or selection programs, that they're or setting up new ones for selection for specific traits. This is both in warm water, cold water, fresh water, and marine environments. So I've got to see a lot of basically with the different life histories you see in all the different fish species and the different types of programs that are set up to manage these in either maintaining broodstock or selecting for traits. There's a lot of variation that occurs. So with that, thank you to Bernd, and I look forward to the panel discussion.
Great, thank you, Ken. Let's have everybody back on the panel, and then we can start our question and answer portion of this webinar. So again, a reminder to all our audience to please post your questions. Our panel of experts are happy to answer your questions. Let's get on to the first questions. Maybe I can, Ken, you can start off with this one. What key changes have you seen in broodstock management in the last 5 to 10 years?
The last 5 to 10 years. Well, depending on what part of the industry you're looking at, whether you're a large production company where you maintain your own broodstock, or you are selling eggs or fry or offspring to producers, there's been a lot better management. I mean, these are becoming more extensive in management of the broodstock, basically limiting inbreeding, following the pedigrees within stocks, and actually following certain traits within the breeding nucleus and also out in production. That's especially following these traits back out in production is probably one of the biggest changes that I've seen starting to occur.
Right. Bert, anything to add on that in that the changes that you've seen in broodstock management for the last?
Yeah, I mean, it certainly depends on the part of the industry. Like in the Perch production industry, I think broodstock management using genetics is wasn't a big thing until recently. I can quickly say how it was for our company. Like until 4 years ago, we really didn't even tag the fish. We had no genetic information, and so all crossing decisions were solely based on the experience of the broodstock manager. We also hardly registered any data. And about 3 years ago, we started to tag the fish. Of course, this then helps a lot to gather information for the individual fish, which then can be used for crossing decisions. But also there, we didn't have genetic information, and all the data gathering was done by hand, manually on PVC slates, writing with pencils. So it's a system prone to errors and time consuming. And so finally, 2 years ago, we made some big steps, and we started to work together with a consulting company like Select. It's a Scottish company specialized in genotyping and genetic consulting. And this basically helped a lot. And then they used the data to produce crossing lists, and from then on we really only made crossings with these crossing lists. And additionally, we started to use electronic data gathering equipment because it's still important. Sorry, the movie is a bit early. The movie is for the next question, I think. Now, what I wanted to say is, it's the data gathering equipment really helps to gain time and to make less errors for data transfers and all that. And I guess including genetic data and this electronic equipment was a big improvement for us.
So yeah, Bert, can you expand on that, especially given the changes that you're talking about? What technologies are you using? I think you have a video to show in terms of what technology you're using in your organization for your breeding
We are
program.
basically using the whole range of the Biomark equipment. So what you can see here, this is the tissue sampling that we are doing. So on the right side is the Broodstock manager. He takes the fish, reads the pit tag. Then there is the electronic measuring board. He measures the fish. Then he puts it on the scale, which is also Bluetooth, and sends the data to a tablet. I'm preparing a tissue with alcohol to quickly clean the fin, and then I take the sample. These capsules are pre-filled with a buffer, and the video is in slow motion, but you can see that the whole process is rather fast. So per fish, it takes maybe 10 seconds or less. The fish are slightly sedated, so with tricain S, so they support the whole process really, really well. So these fish were obviously tagged already, but then it's another perspective. Again, measuring board, really handy, this tool, and the Bluetooth scale. And tissue sampling. There on top, you can see the tablet with software from Biomark to handle all the data. And the whole data is then exported in form of an Excel sheet, and that we can, this Excel data we can then include in our database. And so this makes a really easy and fast data collection.
Hayden, I want to ask you, because you're basically, in your role, you're in touch with all the different types of technologies and the various companies that are using them. What have you seen in terms of development in broodstock management over the last 10 years?
Thanks, Mary Lynn. Yeah, I would kind of echo some of Ken and Bert's sentiment. Certainly from our side, providing technology to aquaculture companies, we have seen an increase in companies, larger companies, becoming more vertically integrated, where they're creating their own broodstock programs, breeding programs in-house. So they're hiring their own geneticists, doing analysis on those sort of things. So the other comment I would make too is on the smaller producers. They are, looking to outside consultants and becoming, more aware of tagging their fish, taking genetic samples, and then utilizing outside, consultants to advise on, which fish to cross with which, depending on what traits they're looking for, that sort of thing. And then kind of, yes, I think we'll get into this a little bit later, but some of the automation of the data collection that Eric mentioned as well. So.
Right. And Ken, I want to get back to you, sort of the same question. And you mentioned this in your presentation around selection for sustainable diets. Could you expand a little bit more on that? Because I think that's sort of like a topic that's really being talked about across the industry, across the board, is sustainability. Can you maybe expand a little bit more on that, the developments that you've made in that selection, what the results that you've seen in selecting for sustainable diets?
Okay, well, specifically, I mean, on our end, looking at it, when we originally started, we had integration of several different stocks of fish. We had about 9 different stocks that we integrated. And our initial crosses on a low soybean meal diet was averaged about 175 grams in 5 months. We noticed that there'd be variation in the development of enteritis, obviously variation in the growth, etc. We followed this along, looking at a number of different parameters. We did histology, we did gene expression, we did some RNA-seq, we did a number of different methods trying to look at what might be occurring, changes in the animals that compared to their performance. What we've found over the past is that with the selection program, we have fish that would not develop enteritis, and we increase the stringency of the selection, and we've done this twice. It's now such that we can have fish that instead of at 170, they're at about 400, 420 grams now in that same time period, and they do not develop enteritis. A really interesting side effects of that is we also looked at pathogen resistance, and we noticed differences in pathogen resistance besides probably being linked to immunomodulation and not development of the intestinal enteritis. We noticed some corollary changes in the ability to passively transport or actively transport amino acids across the intestine, and also how that developed in basically regulation of muscle accretion. So there was a number of different factors that factored in on the selection for this one trait. Other things came along with it, which isn't typical. And as even as Barrett or any of these people, as you select for a trait, depending on the environment you select in and how you select upon it, other things might come in. But specifically in ours, with these fish, they're now actively being utilized by producers. They're, probably in the majority of the germplasm that are reared in the U.S. currently. And again, the goal of that is to reduce the dependence on fish meal. And now the fish meal, feeds have gone from 40% down to about 15%, with formulations varying in the level of specific plant proteins that are incorporated into those feeds. So I hope that gives you a basic background to that.
Yeah, that's really interesting. So I'm gonna get to one of the questions from our audience now. With the exemption of perhaps the salmonids, what verifiable information do you have that the genetic selection programs have increased aquaculture production and production efficiency? Bert, can we start with you?
Yeah, I guess for us, it's a bit early to say. Like I mentioned, we started our whole program less than 2 years ago. And as this will take a couple of years to show the results, I think, we'll have to wait a couple of years. And this is also a tricky part for the investors. They always want to see results quite fast, and so we have to tell them that programs like that, this gives not miracles in 1 year or something like that, that it takes some time, and this is always a bit tricky.
Right. Do you have anything to add, Ken, to that question?
Well, yeah, outside of salmonids, obviously probably the best known programs are in tilapia, where they have the GIF, the GET, and the GO strains. They're utilized, both in Asia, in Africa, and some of those in Egypt. In Korea, they've got olive flounder. In the US, they've got catfish, where they've done extensive genetic selection, and those are released into the industry. And then in Europe, there's been a large concerted efforts in sea bream and sea bass that are beginning to take fruition. So outside of some, there's a lot. And again, with the ability to be able to, mark and manage your broodstock and your breeding nucleus, it leads to, the gains that, it's, I think 20 years ago or whatever else, when it was published, it was shown that gains through genetics, you could be up to 15%. Long term, these are huge once you start these. But the incentive, without being able to actively manage, control, mark, and manage your populations, there wasn't a lot of incentive or improvement on it because it was so intensive and cost prohibitive. Now with the abilities, the sequencing abilities, the SNP chips, and being able to mark and link a trait to a population, a family, or an individual, has led to this increased focus on the improvement in aquaculture.
Right. Hayden, do you have anything to add to that?
I think those guys summed it up very nicely.
So I want to ask you, Hayden, given these technologies at Biomark, what are with the changes that have been happening in broodstock management over the last 10 years? What are you using in your breeding programs, specifically at Biomark? What technologies?
Yeah, so I mean, obviously, we don't have an in-house breeding program, but we focus a lot on our customers' breeding programs, and so yeah, I can speak from the technology side. I guess one of the things that we have seen most is just a focus, as we mentioned, on more on genetics, and then also on the automation of the data collection. So that's something that we have been big on. We've received feedback from aquaculture industry and breeding programs on what data they're collecting, and how we can help them optimize that data and optimize the collection of that data. As there continues to be a focus on the cost of labor, and reduction in the cost of labor. Also the robustness of our data that we are collecting, as Bert mentioned, writing stuff down, trying to write down I'm sure there's people on this webinar who tried to write down a 15-digit PID tag number. I mean, that can be challenging, right? There's definitely some opportunity for some transcription errors. So something, the solutions that we provide, is automation of, taking, when you scan a fish, that PID tag code comes automatically to a tablet, right? If we take a weight, we take a length, we scan our tissue sample with a 2D barcode scanner. So collecting all that data at once, automating it, we can, a lot of processes, you can take what used to take, three people to do, now one person can collect all of that data essentially. So, and we're making that data more robust, which is, in turn, is what we make our management decisions based upon. So I would say, yeah, genetics obviously, and then also just the automation of that collection of that on-site data.
Right. It's certainly interesting to see the evolution of technology and its application into the aquaculture industry. I have a question here. There's a question here for Bert.
Yeah. Okay.
For the European perch, what is actually the growth DPH until harvest size? Size of harvest and average, I guess, feed conversion ratio?
Yeah, harvest size is currently about 180 grams. And yeah, this is something that we also need to, we needed to make a bit of marketing, because the people were used to the perch from the lake, and because the conditions in the lakes were not great during the last decades, the fish were rather small, and the people were used to rather small fillets. So when we started to market these fish at 180 grams, people were thinking, oh, these are not perch, they are too big, this is something else. And so we basically had to convince them, and also the cooks in the restaurants, that this is actually a okay size to, it's better to work with in the kitchen. And so now it is 180 grams for a fillet is about that what we market. And before only the fillet was used, and now we, like I said, we start to use the meat on the back and also on the belly side, and like with the sausage and all the products, this works quite well as well. Feed conversion rate, I'm not 100% sure there. I would have to ask the production manager, but I think it's 1.1, 1.2 or something like that during the grow out phase, but I'm not 100% sure.
Right. There's another follow-up about the perch breeding nucleus. How many families do you have in your stock, and what is the size and age of mature males and females?
Yeah, here I have to say we are really in the beginning of that whole program, and yeah, we plan to restructure a nucleus population. Like I said in the beginning, in the moment we have really 8 different batches that we use, and they spawn about 6 weeks apart. So this allows for a year-round production. And what we do, we really have a closed system that we don't introduce new fish from outside. So we only, when we replace the broodstock fish, we take back adults from the grow-out facility, and this, of course, is not ideal. And it's a danger, yeah, that you lose genetic variability and you increase inbreeding. But fortunately, we saw with our genetic analysis that we are still okay like that. Currently, we take back about 4 times per year adult fish to replace the broodstock fish. But so, like I said, we are in the middle of the development of that program, and yeah, within the next 2 years, the whole Nucleus population will be reduced and restructured.
Right. Let's talk about broodstock selection. Maybe you can answer this one. What are the parameters you look for when conducting broodstock selection?
Well, first off, variation. You need sufficient variation to be able to carry it through long term for your production, and then further on, if you're going to do selection, you want to show that there's variation for the trait under selection, sufficient that you can make the improvements that you hope to see when you have a proper selection index on it. And again, you know, just step back a little bit from what Hayden and Bjorn said. In the past, I mean, people basically just kept large, and by maintaining these large groups, you know, they didn't know, they maintained them as a breeding nucleus, or they took them back out of the production, but they weren't sure of the relatedness between them, between each other. And I mean, they would manage it, they would hope that there was enough variation, but they were not sure of what the crosses were being done. And if they were concerned, they would go outside the population to get some new stocks to add variation in, which again, it precludes any thought of doing selection for a trait. So basically, for your broodstock, you want to be able to maintain that variation, be able to manage your pedigrees, and then if you're going to do selection, you want to be able to basically either maintain your families or mark your families or individual fish, so you know what they're doing, and you can set up a proper breeding matrices to maintain the variation of population you want and make improvements for the trait of interest.
Hayden, do you have anything to add to that?
Yeah, mine's from a very high level and just feedback from, you know, breeding programs that we work with. But two of the main things that I hear are, you know, growth rates, feed conversion ratios, and resistance to disease or parasites. So that's obviously that's a very broad answer, but very on the breeding program side, a very specific question that they may be trying to answer depending on, you know, the environment they're operating in, the species they're working with, so on and so forth. So.
And Bert, for Perch, is there any specific parameters you look for when you're doing broodstock selection?
Yeah, I mean the parameters that were already mentioned, like yeah, related to the growth, like length and weight, and health parameters like disease signs, injuries, and malformations. But very important, as we use the broodstock fish for up to 4 or more years, is the spawning performance. So for the females, we look at the egg quantity, but also the egg quality. And important is also to know the maturation stage. Is it a female that spawns early or late? And because the coordination of the spawning time is very important for the management of the perch. And for the males, we also measure milk quantity, but also sperm quality, like we determine sperm velocity and activity. And this data is really important for the broodstock manager to organize the artificial fertilization and to have a good synchronization of all the animals that he uses.
Right. We're getting a few more questions. There's one here. For non-destructive fish health screening in broodstock, What type of tissue is collected and how frequently is it collected? Is this screening done in every broodfish or just part of the population? Ken, you want to take this one?
So if I understood the question correctly, how do you monitor for disease in your broodstock or your breeding nucleus? Depending upon what pathogen you're interested in. I mean, if it's an internal pathogen such as, going back to Taiwan, it's IHN for a virus, or Flagobacterium. Obviously, you need interior tissue, something like such as kidney. So that's the destructive method of killing. If it's something like sea lice or an outside pathogen that can be taken from skin scrapings, you're looking at that. But typically, you maintain a sentinel population, obviously, throughout your entire production and broodstock that you can screen, and use that to determine what pathogens are present and at what levels they might be. Depending upon the severity and the type of pathogen, this can be either at greater or lower levels, but a sentinel population is pretty standard through most production and broodstock facilities for monitoring that. What's interesting is like what Bernd said, if you're going back and getting broodstock from the wild, not from the wild, I'm sorry, but from your production groups, is that potential of bringing a disease back from your production population into your breeding nucleus, which is always an issue. But what is great about that is that you actually have production trait evaluations for comparisons on how they're performing. So you've got a trade-off when you look at it that way.
Yeah, okay.
Anybody else want to add to that?
Yeah, maybe about the screening, non-invasive methods. We fortunately don't have much problems with systemic diseases, but with the obvious opportunistic bacteria like the Flavobacteria or Aeromonads. And Flavobacteria often cause problems externally, so we regularly do skin swabs and swabs from the gills and determine them on the microscope. When we have diseased fish, then we do a whole analysis and dissection, and we do bacteriology, full bacteriology, and sometimes also histology. I still have good connections to the Center of Fish and Wildlife Health, and so this certainly helps.
Sorry, and just briefly to touch back on that, I mean, there's been a lot of ideas recently to try to look at environmental evaluations to determine pathogen detection, what's in the water, and see if you can quantify that, to use that. The problem is, most pathogens they're prevalent in all the systems, specifically in production, and that's managed through broodstock is to reduce stress such that with the density of your populations you don't have an outbreak. But all that comes back down to water conditions and management. How you can work with that. So there might be potential in the future that by environmental management or environmental monitoring, you can determine levels and link that to either stress or densities of populations for managing outbreaks.
Yeah.
So another question here, in terms of egg quality and viability, is there any nutritional management that you do to prepare your broodstock before entering spawning and during the spawning season? Bert, start with you.
What I can say is that the broodstock fish receive a special diet, but again, one big problem is that there is no specific perch diet. So it's all diets that have been taken from other fish, and what worked best up to now is diets that are relatively low in fat and a bit higher in protein content, because the perch, they really stock a lot of fat, and they immediately also stock it in the liver, and then you quickly see liver damage because of too much fat in the liver. This is a tricky thing because we saw that the only moment the perch really use this fat is when they produce the eggs. So for the broodstock fish, it seems okay if it's a bit more higher in fat, and what the guys do is they enrich the feed with a special fish oil for the broodstock fish. I cannot tell you the ingredients. I don't know it. I would have to ask the hatchery manager. But yeah, this is actually something they do, feed them before they enter in the period where they produce gonads and the eggs. And another thing is, yeah, what Ken mentioned, yeah, important thing is to reduce the stress for the fish, broodstock fish, of course, as much as possible. But yeah, there is a certain amount of stress all the time in the hatchery. This cannot be avoided. And we also started to use immunostimulants before periods when we know the fish will have a bit more stress because they will be handled more often.
Anybody else want to add to that?
Okay, well, like I said, our group, we also maintain a feed manufacturing facility. So we manufacture experimental diets and have produced feeds for a number of different fish species, both broodstock and production and larval stages, etc. And depending upon the species you're looking at, obviously there are oils, certain lipids that are essential for egg production and going into sexual maturation and will link to fecundity and survival in the next stage. But you have to be careful of what your product is, because then there's also fish like there's like what Björn said, when you have high lipid diets, you get fat that occurs in the livers and in the ovaries. So if you have fish like sturgeon, which we have a number of producers we work with too, and they have a whole different way of managing broodstock on that. But their product is caviar. So you want them to grow fast and you want them to have the energy and the proper lipids, but you do not want fatty ovaries, because that is the product of interest when you go to harvest on the eggs. So there's a level where you want it up to a point, but you wanted to limit it, during the egg maturation or, egg development stage. But for most, lipids are the certain essential lipids that are a focus, to these dietary formulations for fish to have better production, higher fecundity, and better survival of the offspring.
Right. And another question here, maybe Hayden, you can take this one. What techniques, because Ken mentioned utilizing matrices. What techniques have the panel seen to help streamline the process of utilizing matrices during spawning? This process has proved to be very time-consuming at our facility. Are there any technologies that are useful to simplify the pairing process using matrices in large populations? Hayden, do you want to take this one?
I'm going to actually turn that over to Ken or Beats.
Okay.
I think they would be able to answer that better than I can.
Bates?
Actually, I didn't understand it correctly.
Okay. So Ken mentioned utilizing matrices. What technologies what technologies can you recommend that help streamline the process of utilizing matrices during spawning? This process has proved to be very time consuming at our facility. Are there any technologies that are useful to simplify the pairing process using matrices in large populations?
I really didn't. I think it's a language problem. I don't get. I don't.
Ken, maybe you wanna?
Yeah. Obviously, that's always been an issue, especially in a large population. What it comes down to is what species you're working with and what you can do. If you can make it so all your fish spawn at the same time, so you know that all individuals are available. All right, there's a number of genetic programs out there that you can use. You can modify them, which most people do in-house specifically for them. But if you can make if you know what's available and you can spawn them all at the same time, and you have, your program set that you want to maintain, this level of diversity in your population, while you want to select most heavily for these traits, you can have your software, your program that you've got go ahead and set up those matrices for everybody that's available. And if you have a marked population, you know who's available, so you know who's survived to being able to spawn. All right. What becomes more of an issue is if you have a strung out spawning season where you have males and females becoming available to use to spawn at different points of the season. All right. Then you only have these fish that go into the matrix at that specific time. All right. But again, your matrix should be set up to, first off, you want to maintain a level of diversity in that. And again, this depends upon the arrangement that you have for your broodstock and your selection program. All right. But you want to maintain that level of diversity in that, and you want to have the highest pressure for that, or at least maintain a selection index for that trait of interest you're trying to improve upon in your population. And again, you can modify those in-house. You can take what's out there and modify it, or there's programs that are available that you can use to manage your populations. And there's a lot of quantitative geneticists that have, Rimmel Blup, that they use specifically, in basically setting up those matrices for the males and females to use for spawning.
Right. So another question is, how do you envision the future of breeding programs given the genetic advancements? Hayden?
Yeah, thanks, Mary Lynn. Kind of some of the stuff that we touched on before. I see a continued push by companies internally, larger companies internally, to continue to develop their broodstock programs, and smaller companies that may not have that capability in-house to continue to farm that out or utilize third parties. Like I think Perch mentioned Select. There are companies out there that are taking samples, working with the breeding program or the breeding manager, setting up some of these matrices that Ken was just talking about, helping them select, for whatever trait they're looking for. So I think we're only going to see more and more of that in the future. I think it's just kind of the way things are headed. Obviously we come from the technology side, the data collection side, so I see more of that being automated, as I mentioned earlier, a reduction in labor rates, more robust data. And another key there too is the more that we automate things, the less we have to handle the fish, right? So anytime we can handle fish less, we're improving our fish welfare, and that's all around good for the breeding program production. So that's kind of where I see things headed in the future.
And Bert or Ken, are there any innovations or future technologies or that you want to, that you're not using right now, that you want to adopt into your own environments, that you're looking at that would make some efficiencies? Bert, let's start with you.
I don't know if we really would need something really new, but we would really appreciate if some services would become less expensive. Like Ken already said, it's been already becoming far less expensive than it used to be, but nevertheless, genotyping and data processing and all that, it's really something expensive. And for smaller companies, this can be really too much. So we in the Perch production world, like it's a growing industry, but it's still a rather small industry. And so for many people I know, this is too much. They cannot afford that. And also for us, it's, we really have to run a really reduced program. We do much less than we would really like to do. Like we basically focus on doing crossings that are ideal, or let's say we really focus on not to do big errors regarding inbreeding, and we would like to keep genetic variability high. But for example, it would be too much for us to run a selection program where we really want to select for individual special traits. And so for us, this kind of services are still too expensive. And maybe we have to hope that all these processes and services will become more affordable in the future.
Right. Ken, are there any emerging innovations and trends that you're seeing?
Oh yeah, there's a couple of really important ones. I mean, first off, if you step back and look at what was done in the past, most people that had broodstock provided eggs, fry offspring basically for their own production and for other producers. It's now, as Hayden said, it's become more integrated. People maintain their own broodstock. They don't sell outside. It's only maintained in-house. But before, when you did that, there wasn't a real incentive for putting a lot of money, time, and effort into genetically improving your stocks, because when you sold them, someone else could take all the effort and time you put into improving your stock and utilize it for them. And basically you lost any gains, and there wasn't any economically. It wasn't essential for that. Now there's basically they use triploidism, basically making them so they're sterile, so you can reuse them. They have all female or all male lines according to typically growth and maturation, but making sterile stocks that you can release to other producers to protect your investment, in the improvements that you've made. So someone can't take your genetic improvement and then make their own brood stock, and basically steal your innovations and your time and money that you put into this. So there's a lot of techniques that are coming out for sterilization of fish, for production fish, that I think is going to be really important. Also, when you look at genetic selection, I realize, like what Baron said, depending upon the value of your stocks and the value of what you have in production, there's only so much you're going to be able to put in, you want to optimize your genetic selection of your improvements and what you can make. All right. So there's other techniques like genomic selection. So you've got trait assisted, or you've got SNP chip arrays that are specific for certain traits that you can use to assist in selecting on traits. Adding that into a classical selection program, it's going to make it a little bit more expensive. You've got more genotyping. You've got to basically evaluate over the array. If you can do that genotyping, at the same time if you want, but that's going to be a little bit more expensive. It's going to be more toward the high end or high value species that are out there. But I see that more and more coming online as that technology becomes cheaper and more available to other species. And again, even to generate these types of arrays, you have to have a significant amount of sequencing background that's been done on your species of interest to begin with, to generate those SNPs, to then generate those arrays. But those arrays and the sterilization are two techniques that are going to be coming up more and more and more as I see it progress here.
Right. So there's we received a comment, I guess, some insights here around inbreeding from one of our audiences. He says, Bruce Swift, we tend to divide into two groups that are relatively distinct. For example, A group and B group. As long as they spawn A to B, we know there are reducing inbreeding, specifically no sibling mating. This is simplistic, but for large production groups, it can work. We tend to be more specific with the crosses at spawning. So that's just a comment from our audience. To Ken, do you use disease challenge rooms? And if so, could you describe the room and how these trials are set? Also, how do you validate induced challenges to those occurring in the farms?
Can you repeat the last part of that question again regarding challenges, on-farm challenges?
Yes, how do you validate induced challenges to those occurring in the farms? So the whole question is, do you use disease challenge rooms? And if so, could you describe the room and how these trials are set? And how do you validate induced challenges to those occurring in the farms?
All right. When we were doing our disease challenges, we were doing that cooperatively with a producer, which was really nice. So they had a setup where we did the disease challenges in small fish, typically about a gram in size at the time, and we would be able to evaluate them for families. We would take in and we would have a certain number of fish and evaluate them for 2 different bacterias and a virus, and we could incorporate that. The problem with the disease challenges in general is they vary. I mean, week to week, batch to batch, that you have what you have for your challenge with, which makes it somewhat tough to replicate unless they've got a lot of experience, which, you know, thankfully, you know, our cooperators have a substantial experience in this. So we would take that information and we can incorporate that. Now, if I'm understanding this right, how do you determine if the disease resistance that you're seeing in your individuals is applicable to what's being shown out on the farm? And you know what? Other people are going to have other opinions, and they're going to be pretty strong in it too, probably. I'm just going to discuss a little bit what we know. Selecting for a specific, I mean, this is really difficult. If you select a specific genoma or a strain of pathogen that goes out there, yeah, you can generate a enhanced resistance to that. The problem with pathogens in general is they evolve so quickly. Or if you do make it more resistant to a specific strain or a specific species, you have to ensure that you're not making it more susceptible to other strains or other species that are out there. So trying to manage that out on the production level, I've seen both positive and negative results occur for this. And I've done probably, yeah, we probably don't want to go too deep into that, but I'll just leave it at that.
Right. We probably have time for 2 more questions. There's one question here. I think, Hayden, you might be able to answer this. Could you explain a little bit more about the measuring board from Biomark, specifically about the precision of the tool?
Yeah, thanks. Great question. So I think we saw it briefly in Bert's video there with Perch, but basically we have a 1/2 meter board and a 1 meter board, and that is plus or minus 1/2 millimeter in accuracy. It has a little magnetic stylus or pen, and basically the fish just goes on it, and you take, you know, fork length or total length, whatever you're looking for. And that is, there's a good shot of it right there. So that unit has its own battery inside of it. It's Bluetooth, it's waterproof. So that's something that we actually built here in-house. We designed it for use on our own projects, and now offer it, you know, to aquaculture companies and breeding programs. So, and then that Bluetooth unit, that will Bluetooth to our Windows or Android-based software app. And so you can have a number of peripherals, whether it's the measuring board, a Bluetooth scale, Bluetooth pit tag reader, that so on and so forth, that all communicates up to the tablet. That way everything's wireless. You know, as we know, working in, you know, a hatchery or an aquaculture setting, you know, everything's wet, everything's loud, it's noisy. So data collection can be challenging. So kind of once again, back to my comments earlier, anything we can do to automate the data collection process, reduce our fish handling time, you know, reduce the amount of labor we need. But that electronic measuring board is certainly a great tool, and we provide a number of those to the aquaculture industry.
And with that, we go to our last question of the day. And this, I think this is a good question to wrap up the whole thing. And because I know we do get a lot of PhD students, you know, graduates that are interested in this field of work. To go around the table, what one piece of advice would you give PhD students or new graduates that are interested in this field of work? Let's start with Ken, then Bert, and then Hayden.
So actually, I've worked with a number of graduate students. I'm on several committees or whatever, etc. Depending upon where they want to go, if they're interested in basic research or going applied or into industry, but no matter what, they should get experience in the field. I mean, out in an aquaculture facility, because what you think you're doing and what they really need to have done or how to approach it without that practical experience, it makes it really difficult to accurately address and you know, have a successful program. but I would suggest that. I mean, obviously go to a good program and someone that will allow you to work with producers during your while you're taking your courses, etc. Will be invaluable for them to make that next step or make that next decision on how they want to apply their degree.
Bert?
Yeah, I basically wanted to say more or less the same thing. It really depends on really the direction the person wants to go. Is it more is he more interested in the practical part or more in the theoretical part? But I also think that internship in a hatchery that uses the latest techniques is certainly really helpful. We saw this when we were starting to work together with Select. They had a lot of experience with salmonids, but none with Perch fish. So until they once went to us and visited us, they really didn't get the whole picture. Like even all the emails we sent them and explanations, nothing beats the practical experience. So I think this is really crucial.
Hayden?
Yeah, I would certainly agree with those comments as well. And, certainly, obviously, as was mentioned, the coursework is great at university, but that on the ground, cutting your teeth, as we would say, real experience, working, whether it's, in a laboratory setting, an aquaculture lab, an actual hatchery setting, with a producer, with a breeding program. I mean, that is invaluable, and that will help you build those skills for the future that future employers are going to be looking for. As an example, one of the lines that's on some of our job descriptions is aquaculture related experience. I mean, we specifically put that on there as an industry provider or vendor, if you will. We are obviously not producing fish or running a breeding program, but we are looking for individuals that have experience in that field so that we can better serve, those customers in the aquaculture industry. So yeah, that any experience you can get is critical.
Right. Great advices. And on that note, we are concluding today's webinar. Thanks again to our speakers, Bert, Ken, and Hayden, for sharing your insights and answering our audience's questions. Thank you.
Welcome.
And also thank you to all of you who joined us today. Again, the recording of this webinar will be delivered to your inbox about 24 hours after this event. So thanks again, everyone, for joining us. Goodbye and stay safe.
Right.
Thank you, everybody.